when it comes to microbial nutrition, the main goal is concept mastery. So for this particular review section, I would want you to think about the major concepts and how we can reinforce the learning in these concepts and master them. So feel free to share with me questions that you may need clarifications on, and I would be glad to answer them as we review them. So when it comes to microbial nutrition, is one of the topics in microbiology which is very relatable because just like humans, microbes also need nutrition to grow. So for this particular one, our main emphasis would be on what are the main things that microbes need to survive and grow when it comes to the nutrients that they need, when it comes to how they can survive within their environment. So if you look at the chapter textbook, it is mostly emphasizing most major nutrients like carbon, hydrogen, and other key nutrients that microbe may need. So hopefully, I'm going to share my screen, and hopefully this would be some of the major concepts that we would mainly review. What do microbes need to grow in terms of nutrition? Now, when you think about us humans, you think about we need carbohydrate for energy, we need proteins, we need some vitamins, we need some minerals, we need water. And similarly, microbes also have needs when it comes to nutrients. So again, recap about macronutrients, micronutrients. So I'm very curious to hear your thoughts. What is the difference between these two? If we say a nutrient is macro, how different it is from micro? Please feel free to share with me your thoughts in the chat section. And can you give some examples of nutrients that are considered macro? So first of all, what is macronutrients and what are some examples? So let's look into the chat just to get some few thoughts, okay? So micronutrients needed in smaller quantities, micro, small. I have another response here. Macro, large quantities. So that is good. Macro is large, micro is small. Another question, can you think about examples of nutrients that microbes need in large quantities? Okay, so I'm getting some response, the schnapps, and I'm guessing that would be carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. Now, these are needed in larger quantities. What is the main purpose or role of these macronutrients? What are their main function? What are their main functions? So if carbon, hydrogen, nitrogen, oxygen, and several other of these nutrients are needed in large quantities, what role is it playing for the microbe in terms of its body? So we'll give a few minutes and see if we get thoughts. Okay. So I'm getting another one in terms of their function. Okay. Any other thoughts about what are their major function? When you think of macro and micro, macro and micro. So I think I have one response here that is for metabolic function. So I'm guessing you're actually probably thinking about the micro. So micro help in enzyme, and then they also help in protein structure. Macro help in the cell structure, and I'm getting another response on metabolism. Yes, definitely metabolism. Any examples of micros? I think I'm getting micro magnesium. Okay, any other examples of macro? So another person also shared carbon. So carbon is needed in larger quantities. Actually, if you have a microbe like E. coli, Escherichia coli, which is very common in the gut of all humans. Now, E. coli reside in the gut, and sometimes it can make its way into other parts of the body where it can cause infection. But if you dry out an E. coli and remove the water, E. coli is made up of 50% carbon, so that is really important to take note of that. Very, very important. I'm getting another examples. Iron is a micronutrient. Now, another question that I want to ask based on the concept you're already familiar with when it comes to microbial nutrition. What is another name for micronutrient? So sometimes some textbook would not say micronutrient, but what is another name for micronutrients? Trace elements, yes, trace elements. So whichever one you see in either of the textbooks, it still means the same thing. Now, these are needed in smaller quantities, not because they are not relevant, but because they have that smaller quantity role to bigger role for the microbes. So when you think about enzymes, enzymes are huge when it comes to metabolism and how energy is generated for microbes. Now, most of the key cofactors and coenzymes are mostly coming from these micronutrient sources. So that is also another important thing to take note about. Okay, I'm still reviewing what do microbes need, right? What do microbes need to be able to sustain themselves and to be able to grow? Now, let's head on to the second concept. Since we've looked at the major nutrients that micro need, macro, micro, we've looked at what are their roles. Now, let's also look at what are the various nutritional types? What are the various nutritional types? So I want to throw out a question out there. Nutrients that must be provided to microbes, what name do we give to that? Because that would help us to begin to look at these types of nutrients that microbes might have. So let's look at some feedback. So nutrients that it is a must, you must have it. So these are essential nutrients. So humans, we have certain essential nutrients, which means we need it for our metabolic functioning, our cellular structure and activity. So it's a must for our survival. Now, the same thing, essential nutrients are a must for microbes. And the 6 essential nutrients are also like the schnapps. So when it comes to the microbial nutrition types, you have to also think of the key essential nutrients like the carbon, the hydrogen, and then you're also going to think about nitrogen is also part of the schnapps. We also have oxygen, there's phosphorus, and then lastly, you have sulfur in that. So the nutritional types, you have to think of each of these. Now, it is important to think about how would microbes get carbon source? From where would they get carbon? From where do you think microbes would get carbon? So let's get your feedback on it. Where do you think microbes would get carbon? What are the 2 main sources that microbes can obtain carbon from? So we'll wait and get some feedback from the chat because I want this to be more interactive. And just like you've been able to read your textbooks and then, watch videos and master the concept on microbial nutrition. Hopefully these questions would kind of interrogate what we know, okay? So I'm getting another feedback that in terms of carbon source, microbes could be heterotroph or autotroph. Now, these are 2 important key terms. So when microbes are heterotroph, where are they getting their carbon source from? When microbes are heterotroph, where are they getting their carbon source from? From organic sources. Fantastic. From organic sources. So definitely autotroph is going to be from inorganic carbon source, which is carbon dioxide. Definitely like that. Okay. Now, also, when you think about the schnapps that we just discussed, I want us to focus mainly on carbon and hydrogen because those are the 2 key ones that microbes really need to be able to survive, right? So in terms of carbon, you are thinking of you having it from organic source or inorganic source, and they can be heterotroph or autotrophs in those cases as well. Now, how about energy? How about energy? So if you look at my slides right now, I'm kind of having a 2x2 matrix where I'm trying to capture how microbes get energy from either light or they can get it from chemicals right here. and then also comparing it to carbon source, either from carbon dioxide or organic source. So please, for this key term, please pay attention to how the huge terms might be considering two nutritional factors at the same time. So you already know autotroph have to do with carbon dioxide, and you already know that heterotroph have to do with organic carbon source. Now, photo means what? Light. So anytime we see the word photo, it reminds us of light. So that means microbe is getting energy from light source. So microbe can get their energy from light through the sunlight, and they should be able to process it through photosynthesis. So a typical example, if you check the textbook, is cyanobacteria. These are photosynthetic bacteria species. So they are able to use sunlight to generate the energy that they need for their own use. And also, when you look at microbes like E. coli, they can make their food, they can get their food through chemical sources. So they are heterotroph and they are chemotroph also because they use chemicals to be able to synthesize their food. Now, you could also have some microbes that are chemo-autotroph. Chemo meaning they are getting their energy from chemical sources, mainly inorganic chemical. Now, if a chemical is inorganic, what does that mean? So I'm curious to know the thoughts for those of you in the chat space. If a chemical is inorganic, what does that mean? Because chemo-autotrophs, they are getting their energy from inorganic chemical source. But what is inorganic chemical? Okay, so we have some few responses. No carbon, no hydrogen. So that means they are getting their energy sources from a source which does not contain carbon and it doesn't contain hydrogen as well. And then chemoheterotroph. So chemo means energy from chemicals. Heterotroph means carbon from organic source. So there's always a combination of this, and it will be a great idea, like you recap your flashcards for these particular terms, and that will be very helpful if you think about it that way. Okay. So I want to move to the next one. So please take note for the essential macronutrients, please review where are they getting carbon from? If it's heterotroph, what are some potential examples of microbes that are heterotroph? Right, so you have Nitrosomonas, you have E. coli, Staph aureus is a heterotroph. E. coli is also a heterotroph. Fungi like yeast or even Candida albicans is also a heterotroph. So these shows different microbes and where they are getting their carbon source nutrition from. Now, also, if you think about the other SNOPs, hydrogen is also key. So let's do a quick review based on what you've read about hydrogen. What are the three key important functions that hydrogen provides to microbes? What are the three key important roles that hydrogen provide for microbes? Three key important roles that hydrogen is always going to provide for microbes. Okay, so they're going to help maintain the pH. So if you think of the word pH, pH stands for potential hydrogen. So hydrogen is really key in determining if a solution is acidic, where there's more hydrogen concentration, or a solution is alkaline or basic, where you have less hydrogen concentration. Now, I'm also seeing other feedback. Okay, free energy. So hydrogen also provides the basic free energy that the body needs. And then lastly, I think I have someone who gave me all three: pH, free energy, and hydrogen bonds. So hydrogen bonds, when you look at microbiology concept on the chemistry of microbiology, there are three key bonds that were studied under that. So there's covalent bond, there's ionic bond, and there's hydrogen bond. Now, hydrogen bond may be the weakest, but hydrogen allows this bond to be formed because you need to have hydrogen first, which is bonding to an anion or any other negative anion molecule for you to get hydrogen bonds. Now, when you think of hydrogen bond, it's very essential. Our DNA, the nucleotide bases are joined together by hydrogen bonds. And is weak because it allows us to easily break it apart when we want to duplicate or make copies of DNA. So those are very essential. So please always remember those concepts, the three key functions of hydrogen for microbes, and similar also for humans as well. Now think about the other macronutrient, which is sulfur. So sulfur is one of the macronutrients, which is also essential. It may not be needed in larger quantities when you compare it to carbon. But why do you think sulfur is important for microbes? Why is sulfur an important nutrient for microbes? So let's see if we can get some feedback also on this. Why is sulfur also important for microbes? Why is sulfur important for microbes? Okay, so let's look at some few responses that are coming in. Because they can create disulfide bonds, and this disulfide bond help to shape the three-dimensional structure of protein. Fantastic. So protein is one of the most complex macromolecules in the body, and it is really important for microbes to have their protein structure intact and for most of their three-dimensional shape in their tertiary structure conformation. Disulfide bond is as a result of sulfur being formed. So that is really an important key. So hopefully we've reviewed concept of the schnapps. Please know the key terminologies that comes with, if a microbe is having organic carbon source or inorganic carbon source. Why is hydrogen important? Why is nitrogen essential for them? And in what form can microbe process nitrogen in their cell? So this particular question, I'm kind of interested in that. In what form do you think microbe can process nitrogen? Because majority of the nitrogen is in the form of nitrogen gas, which is not always accessible. So in what way could we have nitrogen, okay, in the ammonia form, which is NH3? Yes, so that is correct. So nitrogen gas has to be changed to ammonia form for the body or the cell to be able to process it or be useful for it. And nitrogen is important because it is part of proteins. It is also part of nucleic acids, DNA, and RNA. So nitrogen is important. We need it. Microbes also need it in their body as well. And then phosphorus. Phosphorus is also important because it is essential for DNA, RNA, and even when you think of the energy currency of the cell, ATP. Phosphorus is also essential when you think about it that particular way. So phosphates, phosphorus, these are all important when you look at it both ways. Okay. Any questions or any thoughts about those key terminologies or examples? You can feel free to type in the chat section, and then I'll look at them and address them shortly. Any questions that you may have in terms of mastering the concept in the key nutritional sources that microbes need? So we'll give it a minute for any questions that may pop up in the chat section. Okay, so if not, I'm going to move to the next concept, which is the various environmental factors. So if microbes get the nutrients that they need, what are the other environmental factors that could impact microbes? So the environmental factors that could impact microbes. And these factors, oxygen is one of them. There are other gases like carbon dioxide. So those are going to be, I'm going to ask that question very soon. So microbes, so let me put in right here. So in terms of the gas, oxygen is needed, and also carbon dioxide is also needed as one of the gases. Temperature is also important. pH is also important. And osmotic pressure, and as well as hydrostatic pressure. So let's start first with oxygen. Is oxygen gas poisonous to microbes? Is oxygen gas poisonous to microbes? So let's get some feedback on that in the chat. Is oxygen gas poisonous? Because we are all breathing in oxygen, and microbes, which are aerobes or obligate aerobes, are also using oxygen. So is oxygen gas poisonous to microbes? Let's see. Is oxygen gas poisonous to microbes? Any thoughts on that? It could be a yes or no question. No. Okay, most definitely no. So oxygen gas is not poisonous, but oxygen, its interaction with other chemical molecules could be toxic. So for example, oxygen can react with light and create singlet oxygen. And this is a toxic form, especially for microbes that are going through photosynthesis or are using light as a way of processing their food source. So if a microbe, which is photosynthetic, generate a toxic singlet oxygen form, what would be the best way for them to detoxify it? What would be the best way for microbe to detoxify if they generate singlet oxygen, which is an excited state of oxygen based on its interaction or reaction with light? How will they detoxify it? Just to give you a clue, it starts with a C. Carotenoids, definitely, yes. So they need carotenoid. And especially if you look at plants, this principle is very obvious in plants which have an orange color, which is mainly due to the fact that carotenoid is giving it that particular coloration. So oranges, pumpkin, carrots, any plant that you see their fruit is giving you that orange color is definitely a display of their carotenoid and as a way of showing how it's able to detoxify singlet oxygen. Now, photosynthetic microbes that would generate this singlet oxygen, they would also use their carotenoids to be able to detoxify that. Now, especially for us as humans, when we have oxygen as our final electron acceptor during metabolism, what happens to that oxygen after it has finished generating the ATPs that we need? What happens to that oxygen that we are using as the final electron acceptor? It turns to water. So thank you for that feedback. So it's going to turn to water. So what if the oxygen doesn't get turned to water? That is where there's going to be an issue. That means the oxygen is now going to be in the body where it can react and change to toxic forms. Now, if the oxygen doesn't get changed to water, one of the commonest toxic forms which it can generate is SOD, superoxide ions. So superoxide ions. And in our body, we all have an enzyme called superoxide dismutase, SOD enzyme. So I'm just abbreviating it, but its full name would be superoxide dismutase. And you hope that the superoxide dismutase enzyme would be able to break down the superoxide ions completely to water. But unfortunately, the dismutase is able to break down and change this to hydrogen peroxide. Now, hydrogen peroxide is also another toxic form of oxygen. So in this particular case, we need another enzyme to help us. So question for all of you in the chat, what enzyme does the body have to be able to detoxify hydrogen peroxide? Because it's a toxic form of hydrogen. And we have to get the hydrogen being reduced to water, which will be much safer as metabolic water in the body. So what enzyme would the body use to detoxify hydrogen peroxide? Okay, I'm seeing a couple of answers. Hydrogen peroxide can be detoxified with catalase. Catalase, so that is very good. Catalase. So catalase would help break down the hydrogen peroxide completely to water. Completely to water. Now, if this doesn't happen, then definitely you are going to have what we call hydroxyl radicals. And the textbook always emphasized the point that the hydroxyl radical is the most reactive. So it creates what we call free radicals. And these free radicals can cause the cells to go through oxidative stress, where the cells are stressed. And when cells are stressed, various organelles can become damaged. So DNA can become damaged. Proteins can become damaged. And these key molecules, the damage of them could lead to the cell being also damaged where it cannot survive. So it can lead to cell death. So oxygen is very important, and it actually is important that if you are using oxygen, you do have the enzymes to be able to detoxify the toxic forms. Now, there are other key terms here which I'm also interested in. If a microbe is microaerophile, microaerophiles, how much oxygen do they need? Because they also need oxygen. But how much oxygen do they need? If a microbe is microaerophile, how much oxygen do they need in this particular case? So let's see if we can get some feedback in the chat section. So aerobes, they need oxygen. Anaerobes, they cannot tolerate oxygen. Microaerophiles, okay, so I'm getting some feedback. So they only need 2 to 10% oxygen only, just 2 to 10% oxygen. Now, what would happen if the oxygen concentration exceeds 10% or is below 2%? Definitely there's going to be a problem because the 2 to 10 is helping preserve their macromolecules and then their structure for them also. Another question, anaerobes, they cannot tolerate oxygen, or oxygen is poisonous to anaerobes. Why? Why is oxygen poisonous to anaerobes? So let's get some feedback in the chat section. Why is oxygen poisonous to anaerobes? Okay, so I'm getting some feedback. They do not have the enzyme, okay? They cannot tolerate oxygen because they do not have the enzyme. So these are spot on answers when you think about them. And if you can use oxygen, you have to anticipate the fact that there could be toxic forms of oxygen. So you should have the enzymes in the body to break these toxic forms down. So if you look at ulcer, stomach ulcer in this particular sense, I want to give like a clinical application thought right here. So stomach ulcer is typically caused by the bacteria Helicobacter pylori. And this typically causes like peptic ulcer or gastritis when you look at that. And these could lead to an increased risk of stomach cancer. Now, these bacteria that causes the stomach ulcer or peptic ulcer, it requires low oxygen environment within the stomach lining area. So it's a typical example of microaerophile. So H. pylori, which is Helicobacter pylori, is a typical example of a microaerophile, right? Microaerophile. Anaerobes, they cannot tolerate oxygen, and most of them typically might probably lean towards being capnophile. So that is one question that I was trying to bring about, capnophile. So if you don't need oxygen, you might potentially be a capnophile. What are capnophiles? What are capnophiles? Any thoughts in the chat section? What are capnophiles? Capnophiles. Okay, so they likely need high concentrations of carbon dioxide for their growth, right? Carbon dioxide for their growth. So all these environmental conditions, please always think of the key terms and vocabulary. And a flashcard is an important study tool right here, just to keep emphasizing the concept. And then also when you think of some of the examples we are mentioning, it helps you to relate and it helps you to be able to understand them. Now, in terms of temperature conditions, you can have sacrophiles, mesophiles, thermophiles, and extreme thermophiles, also called hypothermophiles. So let me start asking you these questions. What is the temperature condition that is needed by psychrophiles? What is the temperature condition needed by psychrophiles? Psychrophile. So you can just type the number. What is the temperature condition range needed by psychrophile? Below 15 degrees? Yes. So if a microbe is growing at 0 degrees Celsius, it is a psychrophile. 2 degrees Celsius, it is a psychrophile. 5 degrees Celsius is a psychrophile. Provided it is below the 15 degrees Celsius, it is a psychrophile. Now, what type of microbe or pathogen based on temperature can reside with humans in our body? When you look at these four classifications, psychrophile, mesophile, thermophile, and extreme thermophile or hyperthermophile, what microbe do you think can reside in our body? What microbe can reside in our body? Okay, so I'm getting some feedback. Mesophiles. Mesophiles. Now, mesophiles means they have to have close to our body temperature range. So another review question. What is the temperature condition range for mesophiles? Because mesophiles are aligning with humans. So human pathogens are typically mesophiles. But what would you say would be the exact temperature condition range for mesophiles? Mesophiles, 20 to 40. Okay, 20 to 40. And then thermophiles would be above 45. And hyperthermophiles or extreme thermophiles would be 80 degrees Celsius to 121 degrees Celsius. So quick question. When you look at these four classifications and you look at our human body, if you can detect a psychrophile in a human being's body, what would be the state of living of that particular person? If you can detect a psychrophile microbe in the body of a human being, what would potentially be the state of living of that particular human being? So this is kind of like, I want you to critically think through, the different temperature conditions, and then also think about the human body and think about the possibility that can psychrophiles be found if a person is living or in a different state of a person's life. So any thoughts about that? I'll give you 30 seconds to reflect and share your thoughts about that. So if you have psychrophiles, okay, so we have some few thoughts here. So let's see. Yes, definitely the person would be in a hypothermia or death state. Yes, because it's like, death state, the body is mostly preserved cold. So psychrophiles cannot reside in our regular living conditions. You can probably have psychrotolerance, which are mostly between 15 to 30 degrees Celsius, but typical psychrophiles, Psychromona species, which likes in the Arctic Sea area. They cannot reside in human body because they cannot survive the warm temperature conditions. You have some microbes. We have some Coelia species. They are microbes that likes to thrive in near freezing zones. And near freezing zones, you most typically have, a dead person where metabolism is shut off, everything is shut off, and the body is being preserved. That's when you can probably have some of these microbes being able to survive in that particular environment as well. There are Psychrobacter aticus. All these microbes are psychrophiles. They are examples of psychrophiles. They need very cold conditions to be able to survive in that particular range as well. Any questions? So I know pH is straightforward. It's very easy for you guys to think about the classification of pH: acidophiles, alkalinophiles, and neutrophiles. Always think of the pH range. What is the pH range for acids? What is the pH range for a neutral solution? And what is the pH range for an alkaline or a basic solution, right? So always think of that. And then when it comes to osmotic pressure, how would you define halophiles? So let's start from that one. Halophiles. If a microbe is a halophile, how would you explain that? Halophiles. So we'll give a minute to get some responses back on that. What are halophiles? Halophiles. Okay, so we have feedback here. So halophiles like high salt concentrations, high salty concentrations area. And the fantastic thing about mastering concept in chapter 9 in terms of environmental factors is that you can always try to relate the concept. Find different ways to relate. So if something is salty, highly concentrated salt environment, you are likely to see a halophile in that particular environment. Also think of terms like osmophiles. So osmophiles, they like high solute concentrations. Any solute which is in high concentration at all, you will likely see osmophiles in that particular conditions as well. So please always think about examples that can always let you relate the concept to things that are possibly around us. So halophiles, salty environmental conditions, they like that. They like that. So salty lakes, salty ponds, you would see halophiles there. The Dead Sea, you will likely see halophiles there because they have high concentrations of sodium chloride, about 20 to 30% of that within that area. So you need that high amount to be qualified as a halophile. 20 to 30% salty concentration for you to be considered a halophile in that particular area as well. Okay. Any questions about the environmental factors? Any concepts that we need to clarify about the environmental concept? Okay, so in our next review section, we'll focus mainly on concept 4, which is bacterial growth. But for the purpose of today, we just want to master a few concepts. So let's start first. These are NCLEX style questions for critical thinking when it comes to microbiology, microbial nutrition, and growth. So most of these questions, we're going to pause for you to answer them in the chat, and then we'll kind of provide the rationale behind why the answer is this option. So let's start with the very first one. So question 1. So a nurse collects wound specimen or sample from a deep puncture injury. The provider suspects an obligate anaerobe. What action best preserves the likelihood of organism recovery? So you've collected samples from a deep puncture injury and you are suspecting a possible obligate anaerobe. What action can you do to likely speed up the recovery? So A, you place the specimen in an oxygen-rich transport container and send it routinely. B, refrigerate the specimen for several hours before transport. C, use anaerobic transport media and minimize exposure to room temperature. And then D, add saline to the specimen so that the organism can begin log phase. So let's see the responses that we are getting from here. A, okay, so most of you are saying A. So I need you to look at the question one more time and reflect about it. You are taking a sample from a patient. So as a nurse, you've taken a sample from a patient. But before you do the identification, you want to make sure that the sample is well preserved. So look at the question. The question is saying that you're collecting a sample. Before you send the sample, how do you protect the sample to make sure that if you are suspecting an anaerobe, you will likely take it to the provider who is going to do the analysis intact, right? I'm looking at it and some of you are saying A, some of you are saying C. Okay, so in this case, I would go with C because remember, if you are suspecting an anaerobe, anaerobes do not like air, right? So if you are suspecting an anaerobe, the most likely you have to preserve it under anaerobic conditions. so that the microbe is able to sustain itself, right? So you want to do proper identification. So you have to preserve how you are preserving the microbe in anaerobic transport conditions. So in this case, C would be the correct answer choice right here. Now let's go to question number 2. Let's go to question number 2. You have a bacterium, which is one bacteria. It grows along the top of a thioglycolate tube. Which description is most accurate? So remember, we reviewed in our textbook that the thioglycolate tube is mostly used for oxygen. You can use it to check the oxygen concentration of a microbe. Now, if a microbe is growing at the top where there's more oxygen, that means the microbe can need oxygen, right? Now, if it's growing in the middle, that means the microbe doesn't need too much oxygen, neither does it need too less. It just needs the right amount of oxygen. If it's growing at the bottom right here, that means it doesn't need oxygen. Oxygen is toxic to it. Now, in this case, the microbe in question is growing on the top. So let's review your feedback. This microbe is growing right on the top. So what would be the best answer option that you are sharing with me? Okay. So most of you are saying C. So let's look at the answer choice for C. Okay. So this microbe is likely an obligate aerobe because it's growing on the surface of the thioglycolate tube where oxygen concentration is really high. So definitely is going to be an obligate aerobe. So C would be the correct answer choice in this particular case. Now let's look at question number 3. Question number 3. Question number 3. Question number 3. And I intentionally left the keyword fastidious in there just to help us to think about some other possible vocabularies that are mostly questioned in a typical NCLEX style. If a microbe is fastidious, what media characteristic will best support its growth? And here, question 3, there are multiple answer choices. So whichever option you are thinking about, make sure you have multiple answer choices. So let's figure out, first of all, what is a fastidious microorganism? So I'll give a minute to see if we can get some feedback and responses. Okay, so we have a feedback. Okay, so someone is saying C has to be considered in there. I agree. So C has to be considered also. Any other option? 3 of the options are actually needed for fastidious organism, and we would explain that shortly. So C is one. Let's look at the other A. So someone says A, C. Yes, so you need to add vitamins and amino acids. You also need blood serum. There's one more that is needed, which is part of still the options in there. One more categories that is needed. One more category that is needed. So let's see if someone would be able to help us with that. One more category which is needed. E, yes. So they would also need additional nutrients that they cannot synthesize. So first of all, fastidious organisms. So these are microbes that they are complex. Their nutritional composition is complex. And they need certain growth factors which their body cannot synthesize. So A, because they are complex and some nutrients they cannot synthesize, we can begin to look at what are growth factor sources in these answer options. Amino acids, especially the essential amino acids, vitamins, these are growth factors the body cannot synthesize. And this is very typical of fastidious organisms where their complexity doesn't allow them to synthesize these molecules. So they need to be supplied with these from the environment. They also need a serum-rich media just to provide them with the extra nutritional sources, proteins, other amino acids that they need in the serum. And then also additional nutrients, because remember, they cannot synthesize certain nutrients. So any additional nutrients that they cannot synthesize, which they need, is very essential for them. So I kind of saw this question as an NCLEX style question, and I wanted to throw it out there for a typical mastering of microbial nutrition and growth microbiology concepts when you think about it that way. Okay. Question 4, let's review question 4. So you have a hospital patient and the person has developed diarrhea after multiple broad spectrum antibiotic. What explanation best connects the event to microbial growth? So you have a hospital patient, the person has developed diarrhea, and there's been given multiple broad spectrum antibiotics, which means different drug medication that is supposed to kill several microbes at a time, right? Now, which of these questions comes back in? I think I might probably save this question for our next review section because it fits perfectly into our next review questions when we review the microbial growth curve and details. So hopefully you'll be able to join again for that particular microbial review section because this question might be very helpful in that particular sense. So instead, let's go to question 5. Let's go to question 5. So a student states that carbon and energy sources always come from the same place. Which response by the instructor is best? So if I get one of you or any student tell me that carbon and energy can always come from the same place, and the emphasis is always, always. Which of these answer options would you argue to be correct here? Always. Is carbon and energy always coming from the same source based on our review of the key concepts that we've mastered so far? So let's get some feedback. Always, let's get some feedback from here. So which answer option would any of you choose in this case? Okay, so we are having this one. Let's see. So B, okay, I'm getting a couple of Bs, okay. And I agree, the correct answer would definitely be B. This is incorrect. And that is mainly because we explained that, you can get carbon from chemical, organic source, and inorganic carbon dioxide source. So definitely energy can also come from chemicals or from light. So making that huge assumption that energy and chemicals are all coming from the same source is definitely incorrect in this case. So let's look at question 6, question number 6. Question number 6. Which organism would you classify as a chemoheterotroph? Chemoheterotroph. Chemoheterotroph. Okay, so we're getting some feedback. Chemoheterotroph. B, okay. B, chemoheterotroph. Okay. Chemoheterotroph. Okay, so I'm getting another response, which is A. So I will likely go with A because remember, chemo, you are getting your energy from chemicals. Heterotroph, you are getting carbon from organic source. So carbon from organic source. Chemo, you are getting energy from chemicals. So that's how we are going to approach this one. And then you can look through the answer options and then begin to see which one do you have carbon from organic source and then energy from chemicals right here. So definitely you are having organic molecule, which are chemicals, giving you your energy source because they are being oxidized, right? So they are being oxidized to release energy. And then you are having organic carbon also being here. A couple of people also selected B, But let's look at B and look at why that would potentially not be the correct answer choice. So you have a natrifying bacterium. It uses inorganic chemical, and then it has carbon dioxide as carbon source. So these two definitely give it off because heterotroph, you should be using organic carbon source. So this one makes it incorrect. So you can break the key term down and be able to like pick apart which part of the definition in the terminology that is not perfectly suiting the description right here. I think I have two more questions for us to review here. So question 7 and then question 8. Question 7 and then question 8. So let's start first with question 7. So you have a culture that has heavy growth at the top. Remember, again, the thioglycolate tube is used for measuring oxygen, right? With a lighter growth throughout the rest of the medium. Which of these best describe the microbe? Which of these best describe the microbe? So you have heavy growth at the top and you have lighter growth in the rest of the tube right here. So that's question 7, because it looks like it's doing both, right? It is growing at the top and at the same time, it is growing in between at the same time. Okay, so I'm getting feedback here. A, okay, A. And I agree. Remember we said facultative. Facultative is like you're able to do both. You are in one side, but you can do what the other side is also doing. So facultative anaerobes. Facultative anaerobes. They are anaerobes, which means they don't really require oxygen, but they can tolerate oxygen because they do have the enzymes to break it down. So you have some of it growing on the top and some of it growing in the lighter area. So it's definitely going to be a facultative anaerobes. And then let's look at our very last question. So you have a nurse, and this nurse is teaching food preservation. So most of us preserve food in different ways, storage in the refrigerator at different temperature conditions, or refrigerating, or using salt or salt solution brine to be able to store that. So as we think of this question, we'll process it through that way. So you have a nurse teaching food preservation. Which method primarily inhibit microbial growth by creating high osmotic pressure? Select all that is going to apply. So I'll give you a clue here. So for this particular question, there are 3 possible answers, all of which are correct. So when you are picking, try to get at least 3 answer options that are correct in this particular case. 3 answer options that are particularly in this particular case. 3 particular questions, answers that are. So let's see if you can give me feedback responses to that. So A, salting to cure the meat. B, adding concentrated sugar to the jam. C, boiling before drinking. D, you are refrigerating the leftovers. And then C, you are brining pickles. So let's see the options that are here. Okay. A is definitely in there, so I'll highlight it. A is one of them. Let's see. A is one of them, and then we have E. E is also one of them. E is also one of them. And there's one more. There's one more. So I think I'll probably borrow through Miss Sinaya's answer option in my chat, and then go with, I think we have A, E, there's one more. So I picked the E also, and I picked the A also from Sinaya, and then Miles also. Any other one, there's one more missing. So I'm picking the A and E, but there's one more which is missing in the answer choices, which is going to be very appropriate. B, right? So A, B, and E would be the best answer options. Because remember, the question is you are preserving. So you have to think about the preservation. The emphasis is on high osmotic pressure. So in high osmotic pressure condition, the solute concentration has to be higher. Now the solute could be high sugary concentration, high salty environment. So those would be what you'll be looking for. Anything which is concentrated. Those will be the best preservation methods that you'll be looking. And this is a very practical application of, how microbes grow and how we can use them either in understanding clinical application or in food microbiology or in food science and even in food preservation. Because in this case, we can use salts. In this case, you can use a concentrated sugar because it's also a high solute solution. And then brine, which is also having high salt and concentration, can also be used in this particular case. So I hope this section was very helpful for you. Please keep reviewing key terms and concepts. That is the best way to master microbial nutrition and growth. And hopefully I'll see you in our next section when we meet next. So there will be another live schedule section where we would mainly focus on microbial growth. Now, if microbes have the nutrients and the environmental conditions that they need, how do they actually grow? What is the process of their growth? And how can we take advantage of the growth to apply it for clinical application in terms of when do we give medication? When do we treat patients with this kind of information? So I'll see you in my next section, and then hopefully feel free to shoot me any questions